My talk was about the development of the V1 kinase inhibitor. So as the cell progresses through the cell cycle, there are multiple checkpoints built in to prevent cells with damaged DNA to proceed to mitosis and repair the DNA. So V1 kinase results in inhibitory phosphorylation of CDK1 and 2, which prevents the cell from proceeding to mitosis by regulating the G2M checkpoint. So these checkpoints are mainly for the cell cycle progression and are involved in repair of the DNA damage. However, in the last couple of years, there's more information coming out where the CDK cascade is involved in immunity and in T cell energy, clonal expansion, proliferation, development of T cell memory cells. Hence, since V1 kinase inhibitor affects the phosphorylation status of CDK1 and 2, there has been increasing information coming out that modulation of the CDK pathway will modulate the immune response. And so the V1 kinase inhibitor, specifically AZD1775, is being evaluated in combination with an anti-PD-L1 antibody. And the immune modulation is being studied of this pathway. So the V1 kinase inhibitor is currently in phase 2 testing, both in combination with chemotherapy as well as a single agent. And they're specifically trying to evaluate V1 in the setting of p53 mutation positive tumors, since p53 modulates the G1S checkpoint. And so if you dysregulate the first checkpoint and the V1 kinase inhibitor dysregulates the G2M checkpoint, you may result in increased DNA damage, increased cells going into mitosis with damaged DNA. So the main side effects of even single agent V1 kinase inhibition is bone marrow toxicity and effects on the gastrointestinal tract. And that has proven to be a challenge in combining this agent with cytotoxic chemotherapy, since those are the main side effects of those therapies too. So when we conducted the single agent trial of the V1 kinase inhibitor, we saw activity in BRCA positive tumors. So 2 patients out of 8 had confirmed partial responses. One patient had head and neck cancer, the other with ovarian cancer in the germline BRCA background. And so we would like to investigate this agent further in the setting of BRCA germline mutations, as well as patients that have mutations in the other HR repair pathway. So I think the checkpoint, and specifically the V1 kinase inhibitor, is a very promising agent that needs to be developed and looked at both in the context of DNA damage response as well as the immune checkpoint modulation. There are multiple inhibitors of the DNA repair pathway, and how to optimally combine it with the V1 kinase is the challenge going forward. But I think it's a promising strategy in treating cancer.